The overall goal of this protocol is to evaluate both in vivo senescence and reprogramming in the skeletal muscle upon tissue injury.
This method can help to address critical questions in the regenerative medicine field, such as how to enhance cellular plasticity to promote muscle regeneration. The main advantage of this technique is to provide a reliable method to detect and quantify senescent cells in their resident environment.
Cellular senescence has been recently implicated in tissue repair and regeneration. Understanding the mechanisms of how senescence contributes to tissue repair and regeneration will certainly have an impact on regenerative medicine.
We first had the idea for this method when we investigated the potential impact of tissue damage on in vivo reprogramming in skeletal muscle.
To begin this procedure, fix the slides of muscle samples in 1% paraformaldehyde and 0.2% glutaraldehyde in PBS for 4 minutes.
Then, wash the slides with PBS 2 times, each time for 10 minutes. Then wash the slides with PBS pH 5.5 for 30 minutes.
Next, incubate the sections with the X-Gal solution in the dark at 37 degrees Celsius for a minimum of 24 hours. Afterward, wash the slides with PBS 3 times, each time for 10 minutes. Then, post-fix the slides with PBS containing 1% paraformaldehyde for 30 minutes. Finally, wash the slides with PBS 3 times. Subsequently, counterstain with 0.2% eosin by immersing the slides in the eosin solution for 1 minute. Then, rinse them with distilled water briefly,